Welcome to The Kitchen. I'm Cheryl, your Swanky Danky host. In today's episode, we're going to be learning the recipe for turning DNA into the proteins that our bodies are full of. Start with DNA in the nucleus of a cell. DNA has a double helix structure, two strands coiling around each other. The strands have backbones of phosphate groups and carbon sugar molecules that are held together by nucleotide bonds. There are 4 of these nucleotides: adenine A, thymine T, cytosine C, and guanine G. A and T always bind, C and G always bind. This is useful when we only need to copy one DNA strand, but we get the same nucleotide sequences because the same nucleotides will bind anyway. To begin making proteins, we use RNA polymerase II or RNA Pol II, which is an enzyme. RNA Pol II is used to make mRNA, which is similar to DNA but has a different sugar molecule and is single-stranded. Also, instead of thymine, it has uracil to bind with adenine. mRNA is also able to leave the cell nucleus. RNA Pol II will separate the 2 DNA strands and move down, reading the nucleotides on one of the DNA strands, called the template strand. It will pair each nucleotide with their complementary nucleotide, which becomes the mRNA. Once matched together, the single-stranded mRNA dissociates and lags behind the transcription bubble. This allows the DNA to be resealed behind RNA Pol II as it moves on. RNA Pol II stops and dissociates when it hits a nucleotide code that signals it to unbind. This is called a termination signal, and it involves, you guessed it, more proteins. Before it leaves the nucleus, mRNA gets unnecessary genes cut out. The mRNA will then leave the nucleus and move towards ribosomes, which can be found throughout the cell. Ribosomes are organelles that turn mRNA into protein. mRNA will enter the ribosome through one of its 3 sites and be read 3 nucleotides at a time. This is called a codon. Every amino acid has sets of codons that they code for. For example, if mRNA reads GGA, then a glycine amino acid will be brought to it. tRNA molecules bring the amino acids to the ribosome, and they look real funny. The tRNA has an anticodon that will bind to the mRNA's codon to link the amino acid there. Like a recipe, the codons of mRNA instruct what amino acids tRNA brings to the ribosome to build an organism. As more codons are read, amino acids build up in a chain called a polypeptide. When the ribosome dissociates after reading a termination signal, the polypeptide will fold to become a protein. Different proteins depend on the amino acid sequences that it is constructed of, and the amino acids depend on the codons on the mRNA, which depends on the DNA sequence. It all fits. So fellas, you just learned about how your DNA actually becomes you. DNA to mRNA is called transcription, and mRNA to protein is called translation. Together, they are called the central dogma. Pretty legit, eh? See you all on the next episode of The Kitchen.